what is key in that evidence that makes you lean towards that direction? But at the same time, also what prevents you from making a definitive assertion that that actually is the source?
Well, if I could take those questions in reverse order.
Okay.
What we need as definitive evidence is to get access to the records of the Wuhan Institute of Virology to learn what viruses they were working on, what experiments they were doing, and what their results were, all of which have been sealed off by the Chinese authorities, who have also closed down as early as September 2019 the main database for bat viruses. So that's why we can't say anything definitively, and that's the evidence we need probably to make a definitive pronouncement. As to why lab leak seems more plausible to me, one reason is the simple fact of geography. Wuhan is the center of the pandemic, as well as housing the Wuhan Institute of Virology. So the epidemic broke out right on the doorstep of China's leading center for the study of coronaviruses. So that's certainly something that lab leak hypothesis can explain with the greatest of ease. Much harder to explain on natural emergence. Not impossible, but it's very difficult to sort of get the virus to break out somewhere else in China, and then without leaving any trace for an infected person, without infecting his family or anyone else, to jump on a train to Wuhan and then start infecting people there. I mean, it just is a little strained. Another piece of evidence, much easier to explain on lab leak, is the fact that from the start, the virus was very well adapted to humans. Now, the previous SARS virus, SARS-1, took a long time to adapt first to its intermediate host, civets, which are sold in wet markets, and then to humans. So you can trace in minute detail that it makes sort of like 5 mutations to get good at attacking civets, and so another 10 to be able to attack humans just a little, and another 10, I think there are 25 of these mutations in all, before it becomes a real serious human pathogen. If you look at that, if you look to find a similar process in SARS-2, it's not there. Right from the beginning, it's a very good attacker of human cells. Very easy to explain on lab leak, because we know that Dr. Shi, the chief coronavirologist from Wuhan, was working with coronaviruses. She was using them to infect humanized mice. So these are mice genetically engineered to carry the human-type cells in their respiratory tracts. So if you grow viruses in humanized mice, they get to be very good at attacking humans. So easy to explain on lab leak, very hard to explain on natural emergence. And a third point, which is more technical, is that there is a very important element of the SARS-CoV-2 virus called the furin cleavage site, which does not exist in other viruses of its family, which are called sarbecoviruses. No other sarbecovirus has this furin cleavage site. So the usual way in which viruses acquire sort of new genetic properties is by exchanging material with other viruses in their family. What they cannot acquire is some genetic property their family does not possess. So hard to explain how, on the natural emergence theory, how SARS-2 came by its furin cleavage site. Not impossible, but it's yet another difficult thing for the natural emergence theory to explain.